So welcome to the first lecture of Biology 130, everybody. So in this lecture, we're going to be covering the very basics and foundations of genetics. So we're going to be looking at its history, its applications in regards to humans, as well as basic terminology that will lay the foundation for the rest of the course. So of course, the first thing we want to answer is what exactly is genetics? So genetics is a subfield of biology. So as you may know, biology is the study of life. So bio means life, ology means the study of. So essentially, biology studies life. And genetics is a subfield within biology. So genetics focuses on how we go from information, so things called genes and DNA, to a function, so proteins. Also, genetics focuses on how genes are passed from generation to generation. So that definition probably doesn't make a whole lot of sense, especially if you don't have a background in biology. But as we progress through the course, you'll learn how DNA and proteins play a huge role in genetics. We'll also learn exactly how genes and DNA are passed from generation to generation, so from your parents to you to your offspring. So most of you have probably heard of DNA. So most of you probably know that DNA is the heredity molecule of life. So essentially it's how we store information, essentially holds information to make us. However, you're probably less familiar with proteins. So you can think of genes and DNA being information, and proteins are the function. So you think of DNA being like blueprints for making a car, and the proteins, which are another type of molecule, being the actual car itself. So the protein has a function, the DNA stores the information. So this is an underlying theme and underlying concept that we're going to come back to again and again. So DNA holds information, and proteins provide functions. And really, being humans, proteins determine a lot of who we are. So before I go any further, I want to talk about the layout of this course. So there are really four major parts. The first part is DNA to RNA proteins. So we're going to figure out how we go from information again. So that's DNA to proteins, so a function. The second part of the course is going to focus on classical or Mendelian genetics. So this is very old school genetics. So before we knew what DNA was, this is very, very old style of genetics, but it still holds true today. We'll then follow that by molecular genetics. So this is genetics after the discovery of DNA. And finally, we'll finish the course by talking about genetics, policy, and society. So we'll focus on how genetics has influenced human life. And the way I like to think of this course is that it's a tapestry. So certain topics, so I talked about proteins in the last slide, those topics are going to come up again and again. So just like a tapestry has threads weave in and out, topics are going to weave in and out of this course. So you can expect a topic to come back later in the course. So what exactly are genes and DNA? So I've said this a couple of times, but DNA is the heredity molecule of life. And really, DNA is a very simple molecule. It's very big, but it's also very simple. So really, our DNA, or the information, is stored in four different bases. So these bases essentially hold information. So we have A, T, C, and G. So these are actually abbreviations. So in later lectures, we'll actually find out what those stand for. But for now, know that the basis behind DNA's ability to hold information is held in these four bases. So very, very simple. And as you may know, we transmit information in our DNA. So your parents gave you your DNA, and you're going to give your offspring your DNA. So we transmit our DNA. And that's really the definition of heredity. So being able to pass things from generation to generation. In this case, our DNA is the heredity molecule of life, so we're passing DNA from generation to generation. So then what is a gene then? So genes are discrete pieces of DNA. So it's almost like a chunk of DNA. A little bit later in this lecture, we'll go over a more precise definition of what exactly a gene is. But genes, for the most part, dictate what proteins are made in us. And again, proteins determine who we are. So really, we are a product of our genes. So again, genes are chunks of DNA that usually make a protein. So remember, proteins provide the function. Now we're going to talk about a very important concept in genetics, and this is the central dogma. So the central dogma is actually something that's outdated. Despite that, I think it's a good way to frame a lot of what we're covering in the lectures for this week and in the rest of this course. So I talked about in the second slide how we go from DNA to protein, so from information to a function. So there's actually an intermediate step. You don't just jump from DNA to protein. So the intermediate is this molecule called RNA. So that's probably something you've heard of before. So really we go from DNA to RNA to protein. The process of going from DNA to RNA is called transcription. The process of going from RNA to protein is called translation. And again, this course is a tapestry. So absolutely, you'll learn more about transcription and translation in later lectures. So another way to think of this is that we're going again from information to a function. So through those processes of transcription and translation, we're able to go from DNA, which has information, and we're able to convert that information into a function. And that function is provided by proteins. Now I'm going to introduce you to another couple of words. So these are genotype and phenotype. So just like we go from information to function, we go from genotype to phenotype. So genotype is just simply the DNA sequence. You can think of geno, so you can think of gene. So genotype is essentially just the DNA sequence. So in this example where we're dealing with hair color, the genes that determine your hair color, that is your genotype. And specifically, we're thinking about the sequence of those genes. The phenotype, though, is the displayed trait. So your hair color. So again, genotype is the DNA sequence, phenotype is the displayed trait, in this case, hair color in this example. So again, a protein is really determining this. So proteins are playing a huge role in hair color. And throughout this course, you're going to learn about a lot of the roles proteins play in phenotype. So remember, proteins determine the function, and here the function is determining hair color or pigmenting your hair. So again, genotype is the DNA sequence, and phenotype is the trait that's displayed. So both the function and your phenotype are determined by proteins. So this is another way of thinking about the central dogma. So even though your genotype or your DNA sequence or your genes in general play a huge role in who you are, they're not entirely you. So that is, even if you have a twin, that twin is not going to be exactly the same as you. So a twin, even though they have the same DNA sequence as you, they're not 100% like you. So twins, for example, can have different fingerprints, they can have different personalities, and other features. So for example, it looks like this girl on the right has braces, so perhaps there's something different about her that causes her to need braces. So even identical twins are not 100% the same. And this is where the idea of the environment comes into play. So again, we go from genotype to phenotype. However, phenotype is both a combination of your genotype, so your genes, your DNA sequence, plus the environment. And what the environment is, I'll explain in the next slide. But you can think of something like blood type. So if you're A, B, if you're O, you're A, or you're B. So that's determined essentially 100% by your genotype. Your genes determine exactly what your blood type is going to be. So this idea of the environment plays no role in that, or a very, very minuscule role, if anything. So here, your genotype equals your phenotype. This is true for the case of blood type, though. For behavior, it's a little bit different. So perhaps your genes play a role in your behavior. So perhaps your genotype is playing a role. But also, your environment could also be playing an even larger and more important role. So perhaps you have family issues. So that is an example of the environment. Perhaps you'd be starving. You could be exposed to toxins, so pollutants or things like that. So all these are examples of the environment. So again, for something like blood type is determined 100% by our genes or our genotype. For behavior, though, it's a little bit more murky. Perhaps genes play a large role in that, perhaps not. But definitely our environment plays a role in that. So what exactly is this environment? So the environment is really a misnomer. So environment includes things like toxins. So that's probably what you were thinking when you're thinking of environment, but also includes things like how your parents treat you as a child. So that's also considered environment. Also your time in the womb, so what chemicals you're exposed to, how stressed your mother is while she's carrying you, so on and so on. That is also considered the environment. Even things you do to yourself, so smoking is also considered an environment. Essentially anything that doesn't deal with the sequence of DNA is considered the environment. So it's the other factors that influence our phenotype. And this idea that the environment plays no role is hereditarianism. So this is the idea that genes determine 100% of who you are. If hereditarianism was true, then twins would be 100% exactly the same. So the environment is playing some role there. So again, genes do not determine 100% of who we are. Hereditarianism is definitely wrong. But of course, the question is then, how much do they determine who we are? So it's definitely not 100%, but is it 99, 98, 97? Is it 1%, 2%? So this question leads to an existential crisis. So for example, I wanted to eat pizza for lunch today, so I did. So what's determined by my environment? Was this how I was raised? Or what's determined by my genes? So is there something about my genetics, something about how those proteins were functioning? Maybe those proteins were influencing my brain. Maybe those proteins were playing some role in my psychology that made me want to eat pizza. So really, what has been determined by my genes is the question here. And of course, you can then worry that maybe my genes are playing a larger role than I think they are. And of course, you can then wonder, is all my behavior determined by my genes? So has everything been preordained? So is what I want for dinner already decided? Is what I'm going to do tomorrow already determined by my genes? Have my genes already determined what time I'm going to sleep? So on and so on. And this is not too far of a stretch. So if you ever think of dogs, so dogs have a lot of behaviors that are only specific to certain breeds. So for something like a border collie has some sort of genetics that causes it to want to herd sheep or cattle or whatnot. Similarly, something like a Labrador Retriever has some sort of genetics underlying its desire to retrieve. So it's not a stretch then that maybe some of my own behaviors and some of your own behaviors are already determined for us, or there's something driving us in our genetics that drives our decisions and what we do. So this is definitely something the scientists are going to continue to research to figure out how much of an influence our genes have on us and how much the environment affects us as well. So we've talked quite a bit about genes. So here's a very formal definition. So it's a sequence of DNA that codes for RNA and also includes the regulatory sequences of that DNA. And as we saw in the central dogma, that RNA usually then goes on to make a protein. Not always, though. So just sometimes DNA just gets transcribed. So remember, transcription is where you go from DNA to RNA. So sometimes DNA only gets transcribed, but that RNA does not get translated. So there is really not a formal definition of what a gene is. But here this definition is fairly broad and also fairly common in the scientific community. Next, we're going to cover the idea of alleles. So alleles explain how we can be different And really there are two explanations for how we can be different. One is through the environment. So of course we have different environments. We're going to have different phenotypes. So we're going to have different characteristics, different behaviors, different whatnot. So that's explanation number one. So different environments. The second explanation, though, is that perhaps we have slightly different genes. And that's absolutely true. So you know you don't have the same DNA even as your siblings. So you're going to have slightly different DNA even between them. And so these different versions of genes we call alleles. So for example, someone may have a normal hemoglobin allele. So this allele produces normal hemoglobin. So again, alleles are just different versions of genes. Somebody, though, may have a sickle cell allele. So people with a sickle cell mutation in their hemoglobin have major health issues. So here we're looking at two different alleles. We're looking at the normal allele and the sickle cell allele. Both these alleles produce hemoglobin. So hemoglobin transports oxygen through our blood. So still the same protein, still the same function. However, we have a different version of this gene. So here we have the sickle cell allele, and this causes disease. So really, in all honesty, you're probably pretty similar to your sibling as far as DNA goes. However, even small changes in our DNA can have huge influences on disease. So the sickle cell allele is pretty much almost exactly the same as a normal hemoglobin allele. It's only a very small change in the sequence of DNA that causes this disease. So again, our genes are pretty much all exactly the same. However, there can be small and large changes that lead to disease. So large changes like deletions of genes, of course, that's going to cause huge issues for you. If you delete a gene, you're no longer going to be able to make that protein, and that's going to cause an issue. But even small changes, so those changes in those single bases, that can cause a huge issue depending on the gene that's affected. So again, we're all pretty much exactly the same in regards to the proteins we make. But there is some variation, and that variation comes in the form of single nucleotide polymorphisms, among many other things. So let's break this down. So single means one, nucleotide refers to the building blocks of DNA. Poly means many, and then morphisms, so forms. So many forms essentially is what polymorphism means. So a single nucleotide polymorphism, or a SNP for short, are just small single base changes in our DNA. So here we have three different people in this bottom right-hand corner. They pretty much have all exactly the same DNA. So G there, G there, G there, C, 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 A. But here we have a SNP. So here this person's A, this person's G, this person has a T. So perhaps the SNP has no effect on them. So perhaps it's benign. Perhaps, though, this could be a SNP that causes sickle cell anemia. So even though we're producing all pretty much exactly the same proteins, we do have slight differences between us. So even though these single nucleotide polymorphisms, or SNPs, are just one single base difference between different people, perhaps this could change who has a disease and who doesn't. So now we sort of finished laying down the groundwork for a lot of the content we're going to be covering this course. Now we're going to spend some time going over the history of genetics. And it all begins in 1822 with Gregor Mendel. So Mendel was famous for studying pea plants, so he really laid down the groundwork for transmission genetics. So this is understanding how genes or traits go from generation to generation. So Gregor Mendel, if you can tell, the cross was actually a monk. However, in his free time, he did spend time gardening, and this led to his discovery or his understanding of how genetics works, or at least transmission genetics. And this was outstanding because he didn't know anything about DNA, so nobody knew anything about that at this point. So it was just through breeding and breeding of these pea plants that he understood exactly how traits pass from generation to generation. In 1952, we have Hershey and Chase. So they discovered that DNA was the heredity molecule. So again, Gregor Mendel knew nothing about DNA, and it wasn't up until 1952 that people really understood or appreciated DNA as being the heredity molecule. Shortly after that, in 1953, Watson and Crick actually deduced the structure of DNA. So Hershey and Chase just figured out that DNA was the heredity molecule. Watson and Crick actually found the structure. And it was from really these three people, or I should say four, that the foundation for modern genetics was laid down. So genetics has been around for quite some time. And it wasn't really until the 1950s that we really started to understand exactly how genetics works. However, that really didn't stop people from using genetics inappropriately. So genetics actually has a very ugly side to it. And this is in part due to scientists, politicians, and other world leaders using genetics inappropriately. So one of the major events related to the misuse of genetics was the Immigration Restriction Act that was passed in 1924 by Calvin Coolidge. So this essentially prevented Southern European and Eastern Europeans from immigrating to the US, and this was based on them being considered genetically inferior to other Europeans. This was followed up in the 1930s by the Nazis' eugenic movement. So the most basic definition of eugenics is the selective breeding of humans to make them more superior or to make them a stronger race. In more recent times, in 1999, we actually have the death of Jesse Gelsinger. So this is due to a gene therapy experiment. So essentially we're trying to edit his genes. And this led to his untimely death. And his death was really due to the poor practice of the scientist, as well as an immature understanding of the genetics behind how this works. So in 2017, the question is, what's going to happen then? So there are already rumblings in 2017 of genetic experiments that actually may go wrong and may cause huge issues for the people that the scientists are trying to treat. So here is actually a newspaper article published on January 1st, 2017, that talks about the unexpected risks in editing genes. So it's not exactly what they're doing here. So they're actually transplanting things called mitochondria, which isn't exactly editing genes, but still we're influencing our genetics. So you'll learn more about the mitochondria in the next lecture. You'll also be dealing a little bit with this mitochondrial transplant in the discussion board for this week. So even though genetics has a very ugly side to it, there's also some good to it as well. So one example, this is 1990 when the scientist Mary Claire King discovered the BRCA gene. So as you may know, the BRCA gene has been used as a diagnostic test to determine whether or not you're likely to get breast cancer. So here, genetics has played a huge role in diagnostic medicine, and that has probably saved millions of lives. In 2000, golden rice was announced. So golden rice is a genetically modified organism, so it has higher vitamin A levels than regular rice. And this is a huge issue for third world countries where they're not getting enough vitamin A. And 2017 may be promising. So we're still trying to do this gene therapy, gene editing type of experiments. But perhaps now we're finally ready to actually do it while also being safe and doing it effectively. So scientists actually use this CRISPR technology, which we'll actually talk about a little bit later in this course. So definitely, genetics holds a promising future for humans. But it's important to remember all the hideous and horrible things genetics has also done as well. It's important that we remember this so we don't repeat any of these mistakes in the future.